Yeah. Hi, thank you for the invitation, you and Michelle. My name is Mariana Capparelli. I'm from Brazil. I did my master and PhD at Universidade de São Paulo, and there I started looking for study the fiddler crabs, stuff like ecotoxicology and ecophysiology. Now I'm a professor at ICA University, Universidade Regional Amazônica, since 3 years now. And I'm far from the coast, but I study also fiddler crabs here. And now in 3 months, I may go to Mexico to start working the Instituto de Ciências del Mar y Limnología en la UNAM. And now I will start present some kinds of stuff that I did in the last years with fiddler crabs. Who are the fiddler crabs? To start, is the beautiful crabs. No, they are characterized. Let me put it by the asymmetry of one cella in the male. Only the male has the big cella. They are small crabs, range from 1 centimeters to 10 centimeters. They live in this kind of burrows. They are semi-terrestrial crabs. They can live in the sediment and in the water. They are bioturbators. About the diversity. Great diversity on the fiddler crabs are in the tropical and the subtropical areas. Currently, there are 104 recognized species from the fiddler crabs around the world. And the Central America and the South America present almost 54% of the biodiversity of the fiddler crabs. Most studies around the world were initiated by the Crane, Jocelyn Crane, this woman that in the 70s survived for all these countries and started to recognize, identify, and did the ecological stuff with fiddler crabs. There is 11 genera of the fiddler crabs described. 4 genera are found along the Pacific and Atlantic coast of the Americas. In the common genera are the Uca, Petruca, Minuca, and Lepituca. And what is know about the species of Brazil? I came from Brazil. I came from São Paulo, Brazil. And there I started work with the fiddler crabs, but related to ecotoxicological stuffs. There is 9 species of fiddler crabs. The data is new. The area are really well studied with physiological, ecology, and diversity of the fiddler crabs. And about Ecuador, and I came to Ecuador, the data are only from the Crane in the 70s, and the original data from the 70s reported only 40 species of fiddler crabs. And when I arrived here, I started work with fiddler crabs, but different from Brazil. In Brazil, we knew about the species that were there. And I started work there with ecotoxicological, ecophysiological stuff. But here in Ecuador, we saw that there is a lot of stuff to do about the diversity because, like I said, only 40 species were reported, but we survived for all coast of Ecuador, and we found 28 species of fiddler crabs, almost 2 times more. And 5 species that Crane found in 70s, we cannot find anymore. So 25% of the world biodiversity in fiddler crabs are in Ecuador. It's a small country. It's a really impressive number. These crabs are in Ecuador coast, and there is 2 species of fiddler crabs in Galapagos archipelago. And besides this, we survive. So for all the coast of Ecuador in Galapagos Islands, we found 28 species, and 2 species are potential species for the UCN red list because they are found in some particular habitats in this, and this habitat is really threatened by shrimp farms and other kinds of threats. And about Mexico, I'm going to Mexico in 2 months, but there is the fiddler crabs are in all coast of the Mexico. There is 22 species in the coast of Mexico reported, 12 in the Atlantic coast, and the others in the Gulf of the Mexico. And also the data are from the 70s. There is a lot of things to do, I imagine, related to know the biodiversity and the ecological stuffs in the Mexico also, the same way in Ecuador. And so, what factors determine the distribution of the fiddler crabs? The most important factors are salinity, organic matter content in the sediments, the granulometric composition from the sediments, the moisture, the humidity, and temperature. There is some species of fiddler crabs. There are more generalist species that live in all coast and not in all hand of these factors, but there is other species of fiddler crabs. There are specialist species that are restricted in some habitats and needs specifically salinities and the other factors. And be a fiddler crabs, there is a physiological challenge because here is a typical mangrove or estuaries areas, no, that these crabs are living here in this kind of areas. In these areas, like you can see in the picture, are exposed to daily changes in salinity, temperature, immersion, desiccation. So the crabs have to deal with this physiological change 2 times a day, because the tide come and back 2 days, 2 times every day. And also another important physiological challenge for being a fiddler crab, that they live in areas really treated by the humans. Like this is a real pictures, this here in Esmeraldas, Ecuador, the dump area, and crabs living here in this area. Here is a picture from Brazil, people living in the estuaries, in mangrove areas. This is favelas named in Brazil. So the fiddler crabs has the physiological challenge to deal with the all threats like shrimp farms, pool, and in Ecuador it's really common lack of basic sanitation, like this kinds of houses, industries, deforestation. Here I imagine it's a picture from the web, but I imagine like we will see this in Mexico, no? The oil exploration. Just to put in perspective, in Ecuador, this is a real pictures. I and my collaborator Carl, we took these pictures. The Ecuadorian coast lost almost 50% of the mangroves in the last year, in the last 50 years. For example, here is the shrimp farms. This is a mangrove area, and now it's a shrimp farms. Shrimp farms are in all whole coast of Ecuador. Dump areas, fishing areas. Here you cannot see, but this is an island in the coast of Ecuador, Guayaquil. Here there is a lot of plastic containers and anything you can imagine. So why I think it's important to study fiddler crabs, no? Like I said, a lack of recent information on the biodiversity of the fiddler crabs. The data from a lot of countries are from the 70s for the Ukrainian. There is a lot of people studying fiddler crabs, but lack of recent information about the biodiversity of this group. And so the lack of the acknowledgement of the ecophysiology of this species, thinking about these species living in physiological changes. So it's really interesting to understand how these species live in different kinds of environments. There is a lot of species of fiddler crabs, has a wide distribution, and like I said, living in presence in impacted areas and a lot of kinds of impacts. There is some species, there is a sensitive to contamination, so we can use the response to contamination to bioindicators of the ecosystem's health. Sometimes the species live in there in the mangrove in the estuaries areas, and live in contact with the sediment contaminated by metals, by pesticides, by another kinds of contaminants, and they deal with the contamination. So the response of this species to the contamination can help us understand the health of the ecosystem. So I'm trying to study the fiddler crabs in different kinds of evidence lines. Of course, I start the study in Brazil. In Brazil, like I said, there is updated information about the diversity of the fiddler crabs. So there I started with ecophysiology and ecotoxicology stuffs. But here in Ecuador, there is no information about the biodiversity and the ecology. I had to start with the basic information about the ecology, diversity. I did some kinds of stuff with ecophysiology and ecotoxicology. I think in Mexico, I will have to do all kinds of stuffs also, because we always have to understand the ecology of this species, the distribution patterns, to start with the ecotoxicological studies. And thinking about ecological stuffs, it's really easy to work with fiddler crabs. You can do transects with 20 meters like this. This is a transect we did in Galapagos. And in this whole area, we count the numbers of burrows, see the ratio in the male, female, reproductive status, if the eggs in the females, sometimes kept the females and goes to lab and see the fecundity, some kinds of stuff's really interesting to understand, important, This is an example in these kinds of studies that I did, the ecological stuffs. In Galapagos, here is the transectal for Minuca galapagensis. Here is the transectal for Leptuca haileri. Both species are the only species living in Galapagos. Leptuca is the only is a species that there is only there in Galapagos. Is not in the coast of Ecuador. Minuca galapagensis are in the coast of Ecuador. And we see this kinds of parameters that I said, the number of the burrows. We saw that both species has different responses in ecological responses, because they're living in closest, but fiddler crabs, Minuca galapagensis, live in high salinities and low moisture, and Leptuca haileri need the moisture, low salinities, and temperature. So they are different niches, ecological niches, from the both species that live in Galapagos. And about ecophysiological studies, here is the example. It's a metabolic experiments. We put here the kinds of I don't know the name, the circular, this kind of stuff that they put the crabs inside and control the temperature outside, and measure a lot of kinds of parameters. For example, like fiddler crabs living with the wild variation in salinity and temperature, osmoregulatory and metabolism experiments are really interested to understand the responses of the fiddler crabs. So here is an example of the experiment related to temperature. We exposure with the crabs into increasing temperature, and see that crabs are from areas, army areas, have great tolerance to temperature. And here another experiment related to salinity. We put crabs in the range of salinities also, and see some species from more saline areas has more tolerance to higher salinity. It's really interesting to note here, it's a paper not published yet, but the species from Galapagos is a really salt area, more than the coast of Ecuador. The species has this more tolerance to higher salinity. But if you compare Minuca galapagensis from the coast and from the island, the same species from the coast only survived 21 parts per thousand for salinity. And the species from Galapagos, really more, to 64 parts per thousand of salinity. The plasticity in the physiological responses with the same species, but living in different areas. And start talking a little bit about the ecotoxicological studies. There is two kinds of approach that we can do: the laboratory approach or in the field approach. Here it's the examples from pictures from this is Brazil. I did my PhD there. In this area, I collected fiddler crabs, and here there is a most contaminated estuary from the world. There is a lot of industries throw metals into the water in the mangrove area. Here there is a mangrove area that I collected the Minuca hapax, and there is a really contaminated by metals. Here's a pristine area that I compare with the response from the same species. Here is the area in Ecuador that I said there is a lot of plastic. So to do ecotoxicological studies, we have to understand the physiological challenges of the species that we think in work before started do ecotoxicological studies. And so crabs are collected and kept in laboratory for experiments with different kinds of contaminants. This is the approach in laboratory. It's a kind of examples of experiments we did. Here is an experiment did in Ecuador with pesticides. Here is an experiment with metals. We can exposure the crabs to some kinds of contaminants and understand the effects on the organisms. Here is the same experiment here in the pesticides, like I said, but waterborne contaminants here also, but it's like fiddler crabs live in contact with the sediment and feed organic matter contaminated by different kinds of contaminants. It's really important to understand the diet-borne effects of the contaminants. Also, we did these kinds of food for feed the crabs. And for submersion and immersion regimes also. In understand the effects of contamination in situ or in the field, we collect the same species. For example, the work I did in my PhD, I collected here the area of high contamination by metals and compare with another area like this with no treats around by, and compare the response of these species. For example, I collected for my PhD crabs in an area contaminated by methods. I saw that this crabs, Minuca hapax, has high accumulation of metals on these tissues. And another study did here in Ecuador, we collected crabs in that area that I showed. There is a lot of plastic containers, and we can see, for example, this is a picture from the gills, from the crabs, and you can see plastic accumulation in the gills and another tissues. So the fiddler crabs high bioaccumulation of contaminants, has high bioaccumulation of the contaminants. Another kind of experiments, besides the bioaccumulation of contaminants, we can collect in these areas and compare the response, the metabolic and osmoregulatory responses of these crabs, and also changes in oxidative stress enzymes. So I collect crabs in pristine area, contaminated areas. We collect also in both seasons to relate the seasonality and the responses. We saw that crabs from the contaminated areas, there is an impaired consuming of the oxygen and osmoregulation capacity also. And also are really stressful because the enzymes related to oxidative stress are really super expressive. So to finish, there is a lot of issues to follow study. For example, in laboratory studies, how the mixture of the contaminants impact the crabs, the detoxification mechanisms. Like I said, those crabs accumulation a lot of amounts of contamination. Studies like Michelle, I start to doing the transfer of contaminants through the food chain. It's a really interesting population effects on contamination. I think and start understanding these kinds of effects in Mexico. And now in comparing crabs from the areas contaminated in pristine areas, and see there is effects on the population on the population. And like the crabs are bioturbators, if the sediments is the matrix that more accumulate contaminations, no? So it's really interesting to understand the bioturbation with not a lot of contaminants. So I think this is the message I wanna let you guys. Thank you for the invitation. Sorry for my poor English. If you guys cannot understand something, maybe Michelle can help us. If you have some questions, I'm here, and thank you.
Thank you so much. Really appreciate it. And there is a question in the chat. It's from Dr. Martinez Colon. What do you mean by submersion versus immersion?
Yes, because these kinds of this fiddler crabs living in different areas in the intertidal estuary. So this area 2 times today when the tide comes, the crabs are under the water, totally under the water. And when the tide comes out, the crabs are there in the sediments without water. So in the kinds of experiments I said, I try to simulate the tide. So put the crabs totally underwater and put the crabs without water, like they experience in the field. I don't know if I'm clear. I was clear. Michelle, help me.
Yeah, I understand.
You understand me, but I don't know if everyone understands.
Um, could you provide some examples, Mariana, of I mean, you mentioned physiological effects. So what could be some examples of that in the fiddler crabs?
Yeah,
Some examples.
related to contamination, no?
Yes. Mm-hmm.
Yeah. So